Okay, hopefully you had a look at the other lecture on nucleic acids. This one will be specifically for biochem, and we'll go a little bit more in depth on DNA structure. So we're going to talk about nucleotides, nucleic acids, and genetic information. So here are the sections that we'll cover. So we've actually got a lot of material. And we're going to start off with the key concepts about nucleic acids and nucleotides. Again, these are the key concepts that you should pick up as we go and talk. So let's get right into it. So the composition of nucleic acids, there are three parts that we will talk about. Now, nucleic acids are actually polymers known as polynucleotides. So we need to figure out what the individual monomer is, the nucleotide. And there's actually two types of nucleic acids. There is DNA, that hopefully you've all have heard of before. That is deoxyribonucleic acid, and RNA, ribonucleic acid. And each of these nucleotides actually has three parts. It's got a sugar, a five-membered cyclic monosaccharide. That is where the ribo in the name comes from. There is a nitrogenous base, meaning a heterocyclic base that contains nitrogen, and a phosphate group. So we're going to talk about each of those parts. Okay, so the first one, let's talk about the sugar. So in RNA, the sugar is D-ribose, and you can see the sugar here, and yes, of course, you should be able to draw it. Ribose has both of the OHs off of position 2 and 3 down, and in this case, it is in the beta anomer with the OH up, but we're going to see that that OH is replaced by the heterocyclic base. So the key thing to know here is that ribose has both the OHs off of position 2 and 3, and in DNA, it is missing that OH, and that is where the deoxy comes from. And it's indicated in the name, deoxy. Let's go talk about the bases. The bases are generally classified in two different groups, the pyrimidines and the purines. I always have a hard time remembering these, and so I have to think, well, the big name pyrimidine is the small ring or the six-membered ring, and purine, the small name, has the big structure. You can actually see that they are very similar. This pyrimidine portion can actually be found in this purine portion. So really all a purine has is an additional appended or fused five-membered rings with two nitrogens in it. And so you'll hear a lot about pyrimidines. Again, they're the smaller bases, and purines in general. Now, of course, there are specifics. There are the pyrimidines, specifically cytosine. It is found in DNA and RNA. Thymine is only found in DNA, and uracil is only found in RNA. And then you've got just two purines, adenine and guanine, and they're both found in DNA and RNA. And we'll talk more about their structures. And you should be able to draw them. And so when you start looking at these, you realize that they are composed of very similar portions. So like we said, the pyrimidines have this general structure here. In fact, actually what's more common is actually this little structure right here. So it's based off of a pyrimidine, but it also has this C double bond O found in between the nitrogens. And that will be found in all of these pyrimidine bases, C, U, and T. And then what we do to construct them is we have these portions here, which really is just an amine group, or we have a carbonyl group. Now, if we have an amine group, what happens is we include, a double bond in the ring, If we have a carbonyl group, then that nitrogen in the ring will actually have an H. So let's see how that works. So if we take this group here and we mix it with this group right there, so all we do is come in, notice that this nitrogen will match up with that nitrogen, and we just end up with that portion right there. And that is cytosine. And so the way to get the other base is to take the other group, essentially a carbonyl, and when we add the carbonyl to the top, notice that we actually have a NH there. And the reason for that is, of course, we couldn't have a double bond right there, -2, because this carbon would have 5 bonds. So take-home message is when we have that carbonyl up there, the adjacent nitrogen has a hydrogen. So we get rid of that double bond just so we don't break the octet at that carbonyl. If we have an amine at the top, notice with this amine here, it always has an adjacent, or it's connected to the double bond. In fact, it would be what we call vinylic amine. So that's how we get the structures. So again, this is the common group, and either we attach a nitrogen and would include a double bond in the ring, or we attach a C double bond O, and the adjacent nitrogen will have a hydrogen. Now we do have one different structure here, and that's thymine. And thymine differs from uracil in the fact that it just has a methyl tag off there. So in RNA, we use, or uracil is found in DNA. Thymine's found. We'll talk a little bit about that, but you should probably figure out why DNA has thymine and RNA has uracil. What is the advantage of DNA having thymine? Okay, so let's talk about the purine bases. Now, on the purine bases, here is the molecule known as purine, and here is the general structure that we're going to modify. Again, we have 2 pieces that we can put off there. We can either put off of the ring an amine or a carbonyl. And if we put off an amine, remember that it has this adjacent double bond, and that gives us adenine. And then in guanine, we actually have both of these groups there, and that's why we have the and. So at the top, we add the carbonyl. And again, when we have the carbonyl, the adjacent nitrogen is going to have a hydrogen. And then along the bottom, we're going to add the amine. And when we add the amine, that adjacent nitrogen is going to have a double bond. So yes, you should know those structures, and hopefully that little insight and will help show you the similarities, and that really these are composed of just a few different parts. In fact, if we think about it, we've got this part and these 2 parts and that part. And it's really just a combination of those that give rise to these bases. Now again, thymine stands out a little bit because it has that methyl group. But really what you can think is thymine and uracil are essentially the same structure other than that methyl group. So the functional groups off there are the same other than that methyl group, or what we'll say is the pattern of these groups, you know, C double bond O, NH, C double bond O, C double bond O, NH, C double bond O. Those are the same for uracil and thymine. Okay, so start practicing drawing those. And now we've got 2 parts. We've got the sugars, ribose and deoxyribose, and we have the heterocyclic bases. And again, adenine is found in both RNA and DNA, guanines both in DNA, RNA, cytosine, DNA, RNA, and then thymines in DNA and uracil in RNA. And so now let's talk about putting those 2 together. So when you add a sugar and the heterocyclic base, you attach the heterocyclic base at the anomeric carbon. So these are N-glycosides. We'll talk more about sugars later, but this is referred to as a glycosidic linkage. And this is the beta anomer, if you remember that from organic chemistry. And the heterocyclic base is attached to the anomeric carbon of the sugar. And so we get this N-glycoside. And so for RNA, we attach it to the ribose sugar, which has two OHs there. For DNA, we attach the base to a deoxyribose sugar. Notice it's lacking the OH at that 2' position. And so we'll talk a little bit about numbering now. So when they number the heterocyclic bases, they get just the regular numbers. And so you can see that this nitrogen is numbered 1. When we start numbering the sugars, we need to differentiate which way we're numbering, and so they get the 1' and 2' and 3', just to differentiate which ring we are numbering around. So we say that DNA is lacking a 2' hydroxy. That just lets us know that we're talking about the position 2 on sugar. When we say prime, the prime numbers are numbering around the sugars, where the regular numbers would be numbering around the heterocycles. And we'll see those numbers a little bit later. It's not so important to know the numbers around the heterocycles, but very important to know the numbers around the sugars. We're going to see these terms such as 3' and 5' and 2' deoxy. That is a common term. So when we actually attach the heterocyclic base to the sugar, they are referred to as nucleosides. And you may hear that when you hear about nucleoside drugs. And here is just an example of all the nucleosides found in RNA. And I know it's RNA because it has a 2' hydroxy, and these are all of the nucleosides found in DNA. And again, I know it's DNA because there is no 2' hydroxy. So you should be able to tell the difference between RNA and DNA, and that's simply by looking for a 2' hydroxy or lack thereof. Now we go to the complete nucleotide. Now nucleotides actually have phosphates attached. And so you can see nucleosides have 2 of the 3 parts, and nucleosides have 2 of the 3 parts, and nucleotides have all 3 parts. So we can attach a phosphate, make what's referred to as a phosphate ester by taking the alcohol off of the sugar and reacting with a phosphate group. And so it looks kind of like a ether, but they refer to them as esters. I guess you could also argue that this looks like an ester if that was a C double bond O there. So these are phosphate esters, and you can attach them really to any of the free hydroxy groups. What we find is that the phosphates are typically going to be attached excuse me, to either the 5' hydroxy. There we go. That's why we need to know the prime numbering. That is the 5 position of the sugar, or the 3' hydroxy of the sugar. Okay, so that gives rise to the nucleotides when we actually have the phosphate on there, and we've got the full structure. So a nucleotide is a nucleoside plus a phosphate. And that is it. We've got a heterocyclic base, we have a sugar, and we have a phosphate. And then the question is, well, this one right here, what group would it belong to? Where would you expect to find this? And the answer would be in DNA. And I know that because I'm lacking a 2' hydroxy group. So that is a nucleotide that I would expect to be found in DNA. And then the question is, well, which nucleotide is it? And yes, you should be able to identify those and know that this is cytosine. Okay. Excuse me. So when we talk about naming these nucleosides and nucleotides, there's actually some logic we'll see in a second, but you can add more than just one phosphate on there. And those of you that had biology, this will come back and hopefully explain where these terms that you've heard over and over come from. So ATP, now you know the structure. It is adenosine. It is attached to a ribose sugar, and it is a triphosphate. And so you can see that structure right here. Adenosine, triphosphate. No, it's adenosine right there. It's a triphosphate because you have 3 phosphates there. And since I am not indicating that it's deoxy, it's a ribose sugar. If I said DATP, deoxyadenosine triphosphate, then I know it would be this nucleotide right there. And so the difference between ATP, ADP, and AMP is simply the number of phosphates. So triphosphate, diphosphate, or monophosphate there. Anyway, this one right here, this is the least exciting of the molecules. He's kind of damp. So this is a way to understand where the nomenclature comes from. Again, these nucleotides are composed of 3 different things: a sugar and a base and a phosphate. So part of the name, if it is a deoxy sugar, we indicate that in the name. So if we have a deoxyribose, when we start to name the nucleoside or the nucleotide, we start with deoxy to let us know that our sugar is deoxy. Then we name the base, and how we name the base is we combine part of the base name with the type of base that it is. So adenine is a purine, and so we name it as aden. Add OS. I haven't been able to figure out where that comes from, other than maybe it comes from the sugar. In reality, I don't know, but the ene comes from the name purine. You could just say, oh, the ene comes from adenine, but really the ine lets us know that it's a purine base. And you can see for guanine, it's deoxyguanosine. The reason that that ine we know comes from purine base is when we go to the pyrimidine bases, it is named IDINE, so deoxycytidine or thymidine. So the last couple letters lets us know the type of base. And yes, this does seem redundant because we should know that thymine is a pyrimidine base, but the Department of Redundancy department has approved this form of naming. And then finally, if we have a nucleotide, we just add phosphate to the end, and we indicate which position it is attached to and how many of them there are. So let's just read the first example, deoxy. So we know it's missing a 2' hydroxy group. Adenosine, we know that it's adenine and a purine base, and we know that at position 5 we have a phosphate. If I had 3 phosphates, I'd say deoxyadenosine 5' triphosphate. Or if it was to the 3 position, then we would say 3' triphosphate. RNA, the names are a little bit simpler just because we don't have to start with the deoxy, but otherwise they are the same. With the exception that, of course, we would have uracil in RNA and not thymine. And so you can just see where those names come from. I've tried to color code. Again, what we've got is the pyrimidine base in purple, the purine base in blue. In the name, the purple part comes from the base, or the blue part comes from the base. The green part comes from the arrangement of the groups off of the heterocyclic base. So adenine comes because we have that. And then the sugar in orange here, that is where the orange. So the orange part comes from the name of the sugar. The thiamine comes from these groups off of the heterocyclic base. The idine comes from the name of the pyrimidine base. And then, of course, the phosphate comes from the phosphate group right there. So hopefully that gives you a little logic how these names are derived. Okay, there are a lot of other nucleotides. ATP, of course, is one that you've heard of. We'll talk about GTP later on. There's NAD+, NADH, NADP+, NADPH, FAD, FMN. Anyway, we'll see that these nucleotides are actually found throughout biological processes. In fact, one of them is taking ATP, and we actually take that 3' hydroxy group, attack one of those phosphates, kick off two, and we end up with cyclic AMP or CCAMP or CAMP, which is a second messenger, serves to link hormones, first messengers, and enzymes. So we actually use ATP for energetic purposes in our cells as well as a second messenger. And so they say that ATP is a universal energy currency of the cell or some other garbage like that. Really, what ATP is it's a reagent that makes good leaving groups. So it allows chemical reactions because we convert a poor leaving group into a good leaving group, as well as other functions. But it's not like we actually gain energy, like electrical energy or something, when we break those phosphate bonds. And you can see that actually we can get quite a bit of energy by breaking those bonds. So when we hydrolyze it, you release actually negative 45 kilojoules per mole in the cell. So there's quite a bit of energy. That's a pretty high energy bond. You can almost imagine it's like an acid chloride or an anhydride, and so a very reactive molecule. And so what we can do is we can take advantage of this reactive molecule and make less reactive molecules more reactive. And so we couple this hydrolysis of ATP to get reactions to go. So in biology, they always talk about it's like the universal energy currency, and you can imagine like a stock market and people are exchanging ATP to get favors done. That's not how it works. These are chemical reactions, and what we need to do is make it so that we can make and break bonds, and ATP just allows us to make and break bonds much easier. It is a reagent that, like I said before, makes poor leaving groups into good leaving groups and allows chemical reactions to occur at a decent rate. Now, what's interesting is the concentration inside the cells is about 5 millimolar, which is actually a pretty decent concentration. But overall, in the human body, you only have about 17 grams at any time. So if we wanted to harvest all the ATP out of you, we would get 17 grams at any one given time. But you actually recycle your weight or close to your weight in an average day. So if you take your weight of mine, 70 something kilograms. So within a day, I would recycle, not necessarily make, because you convert it from, to ADP back to ATP. So you're not necessarily making ATP, but you're recycling enough. So if you could pull out all those ATPs, you would make your weight in a day. But yeah, let me correct that really, because you're not really making it, you're just recycling enough. So anyway, quite amazing that you would actually use ATP that much within a day. So it is highly utilized. So there are, like I said, other forms that you find these nucleotides when plants make cellulose or starch, you actually add the glucose units together. So here is actually a sugar, and you can see that it is appended to a phosphate. And essentially what happens is this just becomes a very good leaving group, like I mentioned, so that you can actually do chemical reactions. Sometimes you have the nucleotide appended, and really what it is it's just a handle on there. This group really isn't doing anything other than being able to be recognized, but the chemistry is actually happening here. And so this is known as NADH, and the chemistry is actually happening out here. This is a reagent that actually reduces or gets oxidized. So it's involved in redox reactions. And really the adenosine part is a handle that enzymes recognize there, but it's not really playing a chemical role. So our body, or I guess evolutionists, took advantage of these nucleotides and appended other molecules to them to make them useful. Again, we'll get into that a lot. Just understand at this point that nucleotides are not just for DNA and RNA. And here is a summary of them. Difference between nucleosides, nucleotides, difference between RNA and DNA. And we beat that to death. So hopefully at this checkpoint, you can identify the purines and pyrimidines. You can draw the structures of adenine, adenosine, and adenylate, and actually all the other nucleotides. And you can describe the chemical differences between a ribonucleoside triphosphate and a deoxyribonucleoside monophosphate. And when it says that checkpoint, that means in general, you should be able to specifically do that, but you should be able to do it for a deoxyribonucleoside diphosphate and a ribonucleoside triphosphate. So it doesn't mean just be able to do that. It means if you can do that third point there, you should be able to do all other possible combinations. Okay, let's get into the nucleic acid structure when we actually start linking these nucleotides together. And here is a little schematic down in the bottom right, where you can actually see the different level of organization from base pairs to this double helix. We'll talk about these minor and major grooves and how we coil this up into a helix, and then how that helix can actually coil up and coil up even more. Now, RNA molecules are usually single-stranded and form intramolecular base pairs, where DNA tends to have two different strands that base pair by hydrogen bonding. And so we'll get into that, but first we've got to understand how you actually connect these nucleotides together. And so we connect from the 3' hydroxy group to a 5' phosphate there. So what we see is the phosphate is found in between the 5' position and the 3' position. Now, when we make DNA, we tend to make it from the 5' end towards the 3' end. So we're actually adding new nucleotides on in this direction. So when you write out a strand or a string of these nucleotides, you tend to start with the 5' end first. And so if we were naming this, we would start with, and actually I forgot to mention that you should know the one-letter codes, but we would actually list this as TAG. So this trinucleotide here would be referred to as the TAG trinucleotide. Okay, and so we'll talk about actual biological synthesis a little bit later, but you should be able to understand that the backbone of DNA actually goes from a phosphate to a sugar to a phosphate to a sugar to a phosphate to a sugar. That is the backbone. And then off of the sugars, you actually have the heterocyclic or the nitrogenous bases there. But the actual polymer is a sugar phosphate polymer. Okay, so again, DNA and RNA molecules are synthesized from the 5' to 3' group. And so what you do is you actually add a nucleotide triphosphate to the 3' end. So the 3' end is the nucleophile, and you'd attack the 5' end of a triphosphate. And this happens within enzymes, and we need to talk about enzymes before we really get into this process. But just understand that we synthesize from the 5' end to the 3' end. So the 3' end is where it's growing. So when we write out DNA again, it is always assumed that you're writing it from the 5' end to the 3' end. So in this case, this sequence would be an A, U, C, and G. And we're dealing with RNA here. And so that's why it's U. You could have a T there, like it's showing, but then would have to lack that 2' hydroxyl group. Now you can see that there are lots of groups here, and you can actually imagine that this is kind of cumbersome to draw, so they've come up with techniques to kind of make it a little bit easier to understand what's going on. So they have these little schematics down there. Before we talk about that schematic, I just want to point to those heterocyclic bases there, and you can see the numbering. You don't have to worry about the logic of how they decided to number, because it doesn't always make sense. You notice here that they start at U with the nitrogen at 1, and then up at A, the nitrogen to the top left is actually 1. So the numbering of those heterocyclic rings, like I said, we're not going to worry about them. Just understand that's what they get, the normal numbers, and the sugars get the prime numbering. So for example, see how they start right here at the nitrogen that's attached to the sugar, and then 1 is clear up here. So again, don't worry about that numbering. Just understand that the rings, nitrogenous rings, get the normal numbers, and the sugars get the prime numbering. So this little scheme down here, what this is saying is we've got a phosphate at the 5' position. It's connected to the 3', and then this is representing the sugar out there. And then from the 3' position, we're going to another 5' position. That 5' position is connected. The 5', and then this represents the sugar, and then that sugar is connected to another sugar. Essentially, this is representing the sugar and then the base, and this is representing the linkage there. I'm not saying it's clear, but that's one of their ways to look at it. We notice that this is actually a ribose sugar, so this is actually kind of an interesting one. Yes, we know that this is ribose sugar because we have a 2' hydroxy and no phosphate at the 3' position. So again, when we synthesize this, we would actually add a new nucleotide here. And again, we'll get into that synthesis. Right now, we just want to understand that linkage. And again, I'll stress that the backbone is a phosphate, sugar, phosphate, sugar, phosphate, sugar backbone. And off of the sugar is where the bases are found. So we name these generally just by how many of them. So if you just had 2 nucleotides together, you'd refer to it as a dimer. If you have 3, a trimer, a tetramer, or more prefixes. Sometimes when you get larger, you just refer to them as a 10-mer, 17-mer, 75-mer, depending on how many are in there. If you have a few units, they're referred to as an oligomer. Many more, it's referred to as a polymer. So you can write out these short sequences. So for a short sequence here, the P indicates that we're starting with a phosphate on that 5' group. Then you got AUG, G, C, UC. Sometimes when you have DNA, you can put the D out in front, tend to indicate that it's a deoxy. That's often omitted because in most cases you're playing with DNA sequences and not necessarily RNA sequences, but that could get confusing if you're not careful. But just understand that a lot of times, depending on what you're doing, it would be assumed that you're playing with DNA. Here's just another scheme of how it's connected. So like I said, you notice the backbone of this polymer here is a phosphate sugar backbone. Okay, so you should be able to draw those nucleotides together. I should be able to give you some sequence of nucleotides, and you should be able to draw them in the right order from the 5' end to the 3' end. I'd tell you if I wanted a phosphate at the 3' end or at the 5' end, and you should be able to correctly draw out that structure. So when they first started trying to figure out the genetic material, they knew this nucleic acid, and I'll show why. No, actually, sorry, I needed to explain why they call it a nucleic acid. So this phosphate right here, under very acidic conditions, can be protonated. It's kind of like phosphoric acid. At biological conditions, it's deprotonated, but at very acidic conditions, it would be protonated. So that is where it gets the name nucleic acid, because this is actually an acid. It just happens that at physiological conditions, we find it in its conjugate base form. I know that's kind of confusing. Sometimes people look at it and say, well, why is it an acid? I don't see any acidic protons. And that's, well, because we're represented at a pH of 7. It's already been deprotonated. So when they were trying to figure out the structure, they knew that DNA, or they thought DNA was responsible for carrying genetic information. For a while there, they thought it was proteins. Anyway, really interesting story. We'll just keep it short. But there was an Erwin Chargaff that discovered that the ratio of adenine to thymine was 1:1, and guanine to cytosine was 1:1 in a variety of organisms. But then the ratio between adenine and guanine, the ratio varied. So let's just look at a human being. So they just isolate DNA, and then he would analyze, and he would find as many A's as he would T's, and as many G's as he did C's. And then when he compared the A to G, he would find a different ratio, and that ratio would actually vary depending on the organisms. So you can see the ratio between A and G for humans is 1.56, but for E. coli, it's 1.05. And so it actually, that ratio does vary. But for most organisms, the ratio between A and T is 1:1, and between G and C is 1:1. So it wasn't fully appreciated at the time, but this was a key discovery because it should have let people know that there's something that A and T are intimately related. It was really interesting when they first started coming up with the structure of DNA. They wanted to put the sugars on the inside and somehow have those sugars interacting with each other. And that's what gave the information. For some reason, it took them a long time to figure out these bases actually were base pairing and talking to each other. And actually, anyway, this was a key discovery, but like I said, wasn't fully appreciated. So actually, to get to the double helix, one of the key things was some X-ray crystallography of highly purified DNA by Rosalind Franklin. And you can see that picture over here. And if you're an X-ray crystallographer at the time, you would recognize that diffraction pattern as being a helix. Now I just look at it and it looks like, you know, a modern symbol for maybe the X-Men or something like that. But X-ray crystallographers immediately knew that that represented a helix or a helical structure. And so Rosalind Franklin knew that. Eventually it got in the hands of Watson and Crick and was one of the key things to let them know that DNA was actually a double helix. And then you take Chargaff's rules that suggest that G and C would always be somehow related or paired, and that A and T would always be paired. And then there was some other studies in there. Jerry Donahue studied which tautomeric form. Just from organic chemistry, you may actually think that this would be the structure on the right would be the more favored form because you got complete aromaticity. In reality, the keto or the lactam form is actually more favored. So using these pieces of information, and that should be numbered 1, 2, 3, and a bunch of other information and some bad guesses by some very educated people, Watson and Crick ended up coming up with the structure of the double helix. I think it was just one of those aha moments. If you look back into it, Pauling was trying to do this at the same time, and his structure was just really convoluted. It'd really be interesting to get in their minds and figure out why they didn't pick up on the base pairing and the A and T ratio being 1 more quickly. But once they did, the structure came out, was just in a week or so. They pretty much had the structure once they had these key pieces together. So as I mentioned, you may look at that enol tautomer and guess it'd be more stable because you just got those alternating double bonds, but in reality, it was the keto form. So they finally came up with the double helix and realized that it was this base pairing between thymine and adenine that led to a lot of the benefits of DNA as a genetic storage material. And so thymine actually has a complementary base pair set up to adenine. So this lone pair right here can hydrogen bond with the hydrogen on thymine, and these lone pairs on that carbonyl can hydrogen bond to adenine. And same between cytosine and guanine. And you can see that there's a hydrogen down here, and there's no possible base pair. But for cytosine, you've got this, and guanine, you've got this amine here. So adenine just doesn't have any group here to hydrogen bond with that lower C double bond O, but guanine does. So thymine, adenine have 2 base pairs, and cytosine, guanine actually have 3 base pairs. Now, this is just a little sheet that you can use to help you practice these base pairs. Shows how these complementary hydrogen bonding patterns are set up. And it may be good if you actually try to have a CA. See if you can get C and A to base pair, and you'll see that they actually don't. In fact, C and A both have this amine at the top, so those wouldn't base pair. And then they would both have these lone pairs that would repel each other. So that's where we get the fidelity in this base pairing is due to these complementary patterns. And again, this is just a way for you to practice drawing these. And so what I've done is drawn out all the lone pairs and hydrogens here. Here I've just taken away the lone pairs. And here I've actually taken away the groups. I've shown you that there are 3 hydrogens in here. But who owns them, that's up to you to decide. And then for the UA base pair, yeah, figure out who owns what. And there you go. And I don't know why I've got 2 copies on there. I guess just give you lots of practice. So once we get this base pairing, if the molecule just stayed as it was, it would look like a ladder. Now, in reality, what it does is it actually coils up into a helix. Now, since there's two strands, it's referred to as a double helix. Now, you notice that we have directionality on these, where we talked about the synthesis of DNA going from 5' to 3'. But you notice in this double-stranded DNA, the strands are anti-parallel. So one strand is running 5' to 3', the other is 3' to 5'. So you can see that right here. So it's referred to as an anti-parallel strand double helix, and it coils up into a right-handed helix. Now they've got this little mnemonic over here of the right hand, and what it's saying here is if you put your hand here, as you move, your finger should point in the direction that coil goes back. So in this case, your right hand your fingers are following from the palm to the fingers. It's going back. Here, your palms on your left hand would go back. Another way of looking at it is you just follow it from behind, and when it comes out at you, if it goes to the right, it's a right-handed helix. Okay, this is behind. When it comes out at you, it's going to the left. That's a left-handed helix. This is the same type of idea for screws. You got right-handed screws and left-handed screws. And you may think that if you just flip this upside down, it would go from a right-handed helix to a left-handed helix, but it doesn't. And I spent some time looking at a screw yesterday, and it's really interesting. And so what you'd want to do to figure out the handedness of the helix, notice it's coming back. So this is the back. When it comes out here, the helix turns to the right. So I know that is a right-handed helix. Okay, so you want to look at when it comes from the back out, which way is the helix going? And in this case, it's right. And again, it doesn't matter which way you draw it. If you turn that 180 degrees, I promise you it will still be a right-handed helix. Okay, so Watson and Crick first proposed this in 1953. The model suggests a mean for the storage and transfer of genetic information. They realized that you can actually store the information. If I ripped these strands apart, I actually have copies. Now they're complementary copies, but if I rip it apart and then I duplicated one strand, I would have a brand new double helix, and then the other strand, if I duplicated it, I would have another identical helix. So this is a way for genetic information to be carried in this double helix, and it actually offers some error correcting in there. So the strands coil around each other in a helical screw-like fashion. Again, the strands are running anti-parallel, and the heterocyclic bases are on the inside, and the phosphates are on the outside. And again, it's really kind of interesting that it took them so long to figure that out, because it would make sense that you have the charge group, the phosphates, pointing towards water. But those were different times. Not saying that I would have came up with it, but it's really interesting to look back and see the stumbling blocks that people have in science. And once they realize it, it just becomes obvious. So what we have is a 3D structure of DNA, two chains wind to form a right-handed helix. Again, as it's coming from behind and it's coming out at you, notice that it goes to the right, so it's right-handed helix. One of the edges runs 5' to 3', the other 3' to 5'. The bases end up being the core of the helix in here. The sugar phosphates are on the outside, so you can see the phosphates and the sugar groups, they tend to be pointed outward. And the surface of the helix actually has two grooves. So we have what's referred to as the minor groove, so it's smaller, and then you have what's referred to as the major groove, which is larger. It's a little bit hard to see, but this major groove actually runs all the way around, and then it'll appear over here. So if you could twist the DNA around, you'd see that the minor groove is continuous and the major groove is continuous. And so what we have here is a picture of what's referred to as beta form DNA, which is the most biologically common. So just a little bit about the difference in DNA and RNA. In this base pairing, RNA tends not to form these double-stranded helix. They tend to form single strands that do base pair, but they base pair and form loops and stems and all sorts of complex structures that you can see. And we'll talk more about them a little bit later, but yeah, RNA molecules tend to be single-stranded. They can form double strands with DNA, but if you just have an RNA floating around, it will tend not to base pair. Some of that's due to that 2' hydroxy group. And anyway, that just makes it so it's not as stable in the double helix. So, but it does base pair, but you'll just get the short segments that actually are complementary, and then you'll run into a section that doesn't have complementary base pairs to give loops. And so, yeah, RNAs will fold up. So this is just a single strand RNA that folds up into a fairly complex structure. Excuse me. So there we go. There is introduction to nucleic acid structure and the double helix. So we're going to shift a little bit to chapter 24. So if you're following the syllabus, there's some important things in chapter 24 that we need to talk about that go into depth a little bit more about the DNA double helix that will help us understand these nucleic acids. And so off to the right here, you can see the base pairs. Now, I mentioned the minor and the major groove. You can actually identify the minor and the major groove just by looking at the base pairs. So the major groove tends to be what we would say along the top here. And down here are where the sugars are, and then they lead to the phosphates. So the major groove tends to be along the top of these base pairs, and that's why you see the base pairs always drawn kind of the same. And then that top is the major groove, and then this minor groove here is a little bit more crowded. This is where it's linked to the sugar. And again, we'll see that. I just wanted to point that out on the base pair. So when we talk about the major and minor groove, you can actually start to see that in the major groove, this portion of the nucleotides are actually fairly well exposed. So there's actually three main forms of DNA, A, B, and Z DNA. The most common in cells is B form, A you find occasionally, and Z is very rare. In fact, it took them a long time to find the Z form. It tends to be kind of a more dehydrated form, but they have found biological instances of Z in DNA. So if you just stand back a little bit and look at these, you can see the differences. And a lot of times it's the sequence of the bases that actually determines that. But if we look at A DNA, it seems like it's a little bit more squatty. The major and the minor grooves are about the same-ish width. But notice how the bases are actually tilted quite a bit. So B DNA, you've got very distinct major groove. Minor groove tends to be a little bit more narrow, and the bases are kind of flat. And then you look at Z DNA, and the first thing that should stand out there is it's actually a left-handed helix. Again, as we come from out and we start looking, the helix points to the left there. So the most obvious thing for zDNA is it's left-handed. So again, as it comes out and to you, it's pointed to the left, and the out is for the A form of B DNA is pointed to the right. So zDNA is a left-handed helix where A and B are right-handed helix. So now where does this ability to adopt different helical conformations come from? Well, there are lots of different bonds that you can rotate about. So here is the sugar. Now that's a 5-membered ring, so you should remember from organic chemistry that you've got restricted rotation in a 5-membered ring. So really all you can do is pucker there. But all these other bonds you can actually rotate about. Now when they're saying rotate here, you got restricted rotation that just, again, allows puckering. But at these others, you got kind of full rotation. So here is the glycosidic bond. So here's your nitrogenous base right there. And so you can rotate around there. In fact, let's just go down and look at some examples here. So we got adenosine here. And so you can see that you could get syn-adenosine or anti-adenosine. And so that bond is referred to as chi. So the chi gives the orientation around that glycosidic bond. Now pyrimidines, remember pyrimidines are sorry, just a second. Yeah, so pyrimidines, there we go. Got distracted for a second right here. It actually doesn't, these don't undergo syn conformations because this would bump into the ribose sugar right there. So the pyrimidines, so T, U, and C aren't going to undergo this syn addition. Now, since I've got a 5-membered ring right here, notice that it actually kind of tilts the big bulky portion up. So you can get syn-adenosine right there. But just in this 5-membered ring, if I tried to put that C double bond O over here, I would get some steric clashing right there. And so we do have different ways that we can rotate around the bond. We can rotate around that phosphate. The take-home message from this is you don't have to know all the Greek letters for these torsional angles. Just know that DNA has actually got quite a bit of conformational mobility. And in order to get into a very organized double helix, we've got to restrict that mobility somewhat. So if we actually look down the axis, so we're looking from the top down on alpha, or not alpha, sorry, A, B, and Z DNA, you can actually see that there's quite a bit of difference there. So in A DNA, you actually got quite a big hole. Now we talked about the major and minor groove, and you can actually see that, like I said, by looking at the bases here. So this is actually the major groove, and I know that because here is the anomeric bond and the sugars, and so that's going to be the minor groove. So the major groove kind of points you got the axis of the helix here. If you look at B DNA, the axis of the helix actually runs right through the center of the hydrogen bonds between the base pairs. And then you look at Z DNA, and the axis is actually closer to the minor groove. And again, we can tell that because here's the sugar, so you get kind of the minor groove that's more towards the middle there. So a lot of this has to do with sugar pucker. So we talked about you got syn and anti-conformation of the heterocyclic base, but you can also have sugar pucker. And so they've got these C3 prime endos. So it's prime, so we know that we're talking about the position of the sugar. Now, if you remember from when we talked about Diels-Alder reactions, we had endo and exo. And so if we had a bicyclic compound here, then this would be referred to as endo, and exo is more outward. And so in this case here, we've got 3' endo, and what that does is it actually pops up the nitrogenous base there, and you can actually see that it puts the phosphates quite close together. When you do C2 endo, right here, what it does is those phosphates actually go a long ways away. So if you look at C3 prime endo, you can almost say that those phosphates are syn to each other. They're kind of both diaxial. And then if you go to the C2 prime, then you got one axial and, or sorry, they're both diaxial. Let me try that again. Oh my goodness. I'm having technical difficulties, so I'm kind of frazzled. So C3 endo, what it does is essentially puts them cis and pointing in the same direction, puts them in close proximity to each other. When we go to C2 endo and we pucker that ring, those phosphates are a long ways away. It's hard to see in here, but the conformation of the sugars changes quite a bit, and that's part of what's causing these different conformations. And I'll have a slide later that shows that a little bit more. What I want to do is talk just a little bit more about the major and minor groove. And so up here in this structure right here, what we've got is a picture of beta DNA, and you can see that the major groove is a lot more exposed. And what they have here labeled is acceptor, donor, acceptor, 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 donor. So what happens when you get this base pair, you get the base pair between kind of the faces of adenine and thymine, as you can see over here. But you've also got a lone pair here, you've got another hydrogen here, you've got another lone pair there, and then you've got a methyl that's not going to do anything here. So this is the major groove, again, across the top there. And that is exposed in a fairly open region. And so biomolecules will take advantage of this hydrogen bond pattern and will recognize these bases here. So not only do bases recognize bases, but other biomolecules can come in kind of to the other side and recognize this hydrogen bond pattern. Now, this isn't the hydrogen bond pattern between adenine and thymine. This is the hydrogen bond pattern across this major groove, this exposed surface. Now, you can see in the minor groove, at least in beta DNA, it's actually a lot more hindered, but you're looking across the bottom here, and again, you can see that there are places to hydrogen bond. So this is really interesting because DNA can be recognized by other molecules like proteins, and you can essentially tell what type of nucleotides are being base paired inside by base pairing to the outside there. So normally we just talk about the base pairing here, but the base pairing along these faces is also very, very important. So I've always found this fascinating because you don't just use the base pairing to keep DNA together. You actually use other faces along those nucleotides to recognize DNA. And so again, if you look at A, B, and Z DNA, you can see that you get different exposures of these major and minor grooves, which allows different access to recognize these base pairs. And so what you can do is you can come along here, and if you find the right hydrogen bonding pattern, then you know what the sequence of that is. I don't have to open up the DNA to figure out what the base pair is. I can actually read along the outside here and read the hydrogen bonding pattern, and I can actually tell what that internal sequence is. So if we compare A and B DNA, I'm not one to have you memorize a lot of numbers. So if I ask questions on this, I would probably give you the numbers, but there's probably a few that you should know, and that's specifically about DNA. Like I said, A seems to be a little bit more squatty, so it actually has a wider diameter. B, D, and A is a little bit skinnier. And then you should know the number of base turns per helice. So for A DNA, it's 11.6. For B DNA, it's 10. It's actually a little bit closer to 10.4 or 5, I believe. But again, this can vary depending on the conditions that you're in. The helical twist, that's how many degrees you're twisting for every base pair. And so really it's just a calculation between the base pairs per helical turn. And so if you notice that for the bDNA, each base pair twists 36 degrees, you've got 10 base pairs, and so overall you're going to be 360 degrees once you have 10 base pairs there. Okay, so you can see those differences. So for bDNA, we should probably know that there's 10 base pairs per turn, which means that each base pair, again, is twisting at 36 degrees. The helical pitch is the rise per turn. So essentially, every time you turn, how far have you progressed? You notice for aDNA and bDNA, it's the same. But aDNA is a little bit squattier, but it includes a few more base pairs per turn. So you still, each turn, you only progress 34 angstroms, but you've got a few more bases in that distance. You can see that for aDNA, the bases are tilted from the axis quite a bit, where for bDNA, not so much. You can see a description of the major and minor grooves. And again, you notice that the sugar pucker actually varies quite a bit. And also the glycosidic bond formation, they're all anti. Okay, when we look at zDNA, we start to see some interesting things. So this is just a comparison, the pictures between bDNA and zDNA. But the helical sense for zDNA, it's left-handed. The diameter, it's much skinnier. Base pairs per turn, 12. But it's interesting, it's kind of in what they refer to as 6 dimers, because the pyrimidine purine steps and the purine pyrimidine steps are different. So you actually have to look at this in kind of a group of 2. And so overall, for every 2, you're turning 60 degrees. Again, don't worry so much about that. Just understand that it's a left-handed helix. What's really important, or not important, but interesting, is look down at the sugar puckers, and you'll see that the pyrimidines actually pucker different than the purines. And also the glycosidic bond is different for the pyrimidines and the purines. So the zDNA, you've got a lot of different, well, not a lot different, but you've taken advantage of those different conformations. And so you've got syn for the purines, so they're actually kind of pointed back over the sugars. You got 2 different types of sugar puckers in there. And again, we tend not to find zDNA as often, but there are examples of it existing. So zDNA, it actually took, like I said, a long time to find it. They first discovered it when someone crystallized it out of a high salt content. And if you have sequences where you've got lots of GC sequences or ACGT sequences, so essentially what you've got is lots of GC, GCs, GCs, or AC, and then GT, then you'll actually start to get the zDNA. And so, like I said, down here at the bottom is a lot of it has to do with the difference in sugar pucker. So again, just understand that there's a difference between A, B, and Z. Do know that Z is left-handed. Do know that it takes advantage of different sugar puckerings. Know that bDNA is the most common, and that how do we get rise to these different conformations? And so it comes down to the sugar puckering and the orientation of the purines and the pyrimidines. So here's just a picture showing that they actually found a protein that actually complexes with zDNA. So they have found this little motif. They refer to it as the Z alpha. and they just happened to find some proteins. And again, these cartoons may not make much sense because we really haven't talked about proteins, but just understand that at first this was just an experimental curiosity for zDNA, and then they did find it does exist. Okay, here's just a summary of these forms. I've given you kind of a different picture of what endo and exo does. And again, you can see that this endo puts these phosphates in different directions. C2 endo or C3 endo, these are much closer together. Okay, so here is just another picture. I like this picture. You can see things a little bit better. You can see that A is kind of squatty, B is just your typical, doesn't that just look like a nice, pretty helical structure, and zDNA just looks like a mess, really narrow. And anyway, lots of different conformations of, like I said, the bases as well as different puck cream and the sugar. So we're going to pause right there. Sorry that was somewhat maybe disjointed at times. Technology just about killed me on this. Hopefully it's making sense. Now the next section, what we're going to do is we're going to talk about supercoiling of DNA. And this is best represented, I think, by using some models. So it won't be a lecture from me, but I'll have some YouTube lectures that do some really good models to explain these next couple sections. And that should get us caught up. I'll have the next lecture posted before Monday morning, and that will get us back on track. We do have, you know, a flex day. We do have the possibility of moving the test back to Friday, but I think we can get through this. I'll hopefully get the structure of the forces that stabilize nucleic acids up shortly as well. Anyway, thanks for hanging on. If you have any concerns, I haven't heard from any of you. I'm going to start setting up times where you guys can check in with me, actually make an assignment so I can actually see some students and talk to someone besides myself. But yeah, stick with it. Make sure you're learning, bring questions to me. This is new for both of us, so if you have concerns, make sure you bring those up and we can work through those. Thanks. Bye.